So the first speaker for our three short presentation is Dr. Michelle Kahlenberg. She's a professor in the Division of Rheumatology in the Department of Internal Medicine. She's one of our star immunologists here at the University of Michigan. And she's going to be speaking on when inflammation hides, when inflammation hides in plain sight. Michelle?
Thank you, Steve. So I think this worked out actually fortuitously because you gave a very lovely presentation on why blocking interferons might be bad, and I'm going to talk about why interferons are bad. And also I apologize for my title. I didn't mean to steal the same one. I must have read your title and then slept on it and then turned this one in, but I think it actually, it works. So, and you'll understand why I have this sunbathing picture here in a second. So, my lab works on systemic lupus, which is a systemic autoimmune disorder that really affects many different organs. And we think it's driven by a combination of genetic susceptibility and environmental triggers that really drive enhanced innate immune responses, and then this leads to autoreactive T and B cell activation. And really, it's been an intense area of research, really looking to find better treatment options for our patients. And one of the things that fascinated me when I first started learning about lupus was that skin disease is really common in these patients. About 70% of all patients with systemic lupus will have skin inflammation as part of their disease. And sometimes it can actually be quite refractory. We can get their lupus nephritis to settle down, but their skin will continue to misbehave. And the other thing that's very interesting about the skin disease in lupus patients is that skin flares that are often triggered by the sun can often lead to systemic flares. So we kind of see the sequence. The skin acts up, and then the rest of the lupus will follow. The other thing that's very important for our patients is that cutaneous lupus flares are a real major contributor to a lower quality of life. It raises the rates of unemployment for patients, and it doubles the cost of healthcare when their skin disease is active. So one of the things about lupus skin is that it's very photosensitive. So we think about this as sensitivity to either ultraviolet light from either the sun or other sources. And for autoimmune patients, we define this typically as having feelings of illness or not feeling well and getting rashes after sun exposure. And this is what a photosensitive lupus rash can look like. And for significant exposures, like I said, this can sometimes lead to systemic flares of disease. And when surveyed, up to 93% of lupus patients will report photosensitive responses to ultraviolet light. And there's this interesting connection between UV exposure on the skin and the development of systemic disease. And so this is just an old paper from 1985 where they had looked at lupus-prone mice and exposed them to a high dose of UV light. Let's see if my pointer will work. And you can see that some of the mice in this model, the BXSB male mice, have an extra copy of TLR7 and are more prone to disease. They died very quickly after exposure to UV light. And this was because of development of kidney inflammation. And we see the same thing in human patients. And so this is just a case report of a lupus patient who went out and in the sun and developed a very severe case of lupus nephritis, and you can see the rash that followed. And also there's case reports of things like myelitis, which is inflammation in the spinal cord, also that are triggered by UV exposure. And so one of the questions that I was really interested in tackling when we started our work was, why does UV light, which we normally think of as an anti-inflammatory stimulus, we treat some skin inflammatory diseases like psoriasis with UV phototherapy, why does UV light make lupus patients sick? And as part of this, is the healthy appearing skin in lupus really healthy? Is there something about it that predisposes to inflammation? And so in order to understand more about this, you have to understand the structure of the skin itself. And we will focus in today on the epidermis, which is the top layer of skin, made up of keratinocytes. There's a basal layer here, and then these differentiate and slowly lose their nuclei and become a bag of proteins and lipids that forms a very nice skin barrier at the surface. And importantly to know, it's this basal layer of keratinocytes that absorb almost all of the UVB that hits the skin. And another interesting thing about keratinocytes is that they're really poised to communicate. This is a review that we wrote with Johanka Johnson where we just highlighted all the different chemokines and cytokines that keratinocytes can make when exposed to various triggers in culture. So one of the things we wanted to ask was, how do keratinocytes from healthy appearing skin from lupus patients behave? And so we took biopsies from the upper thigh, which is typically a non-sun exposed, non-lesional area in lupus patients, and compared them to biopsies from healthy control patients taken from this same location. And what we found was that there's a really high rate of a type 1 interferon called interferon kappa being chronically expressed in lupus keratinocytes. So this is a lupus looking at interferon kappa, and the other thing that you see is that below that, STAT1 and STAT2, which are downstream signals of interferon triggering its receptor, are also chronically upregulated. We also see that we can capture more interferon kappa being secreted from keratinocytes after UV exposure in culture. And nicely, other people have also shown that this is in fact true. This is a UVB photo testing in human lupus non-lesional skin that was done by Ed Vital's group, where they showed by RNA-scope that they could also see upregulation of interferon kappa. This doesn't project super well, but trust me, it's there. So we think that interferon kappa is being made. We also then used single-cell sequencing to try to understand what populations of the skin really are making a lot of this interferon response and production. This work was led by Allison Billy, who's a junior faculty in dermatology here at the University of Michigan. And one of the things I want to just focus you in on, this is a UMAP of the keratinocytes that we got from skin biopsies from either healthy control, which is the pink, non-lesional lupus skin, which is the green, or lesional lupus skin, which is the blue. And you can see these are all keratinocytes. The basal keratinocytes are located here, and the basal keratinocyte population has a special population that looks more inflammatory in that little dot there. And so that inflammatory population is made up primarily from non-lesional keratinocytes from these lupus patients. And when we applied interferon signature scores from various cytokines onto the keratinocyte UMAP, what we found was that this basal inflammatory keratinocyte population really had a very high interferon score, suggesting there was a lot of interferon signaling going on. So confirming the work that we had seen in culture directly from these cells from patients. We've also shown that these basal keratinocytes that we isolate from non-lesional skin in lupus patients also have a much bigger interferon response when compared to keratinocytes isolated from healthy controls. And this work was led by Alex Choi and Grace Heil. And these Manhattan plots show the bulk RNA-seq effects of treating either lupus or healthy controlled keratinocytes with the indicated type 1 or type 2 interferons. And these Manhattan plots, the higher the signal goes, the more upregulated the genes are. And I think you can get a flavor that the healthy controls have upregulation in response to interferon, but the lupus patients are just much more robust. And Alex's analysis really showed that there was about 121 lupus-specific interferon genes that seemed to be heavily skewed in these keratinocyte populations. We've done a lot of work trying to understand what are the functional consequences of these high levels of interferons that are produced in non-lesional lupus skin. And I just summarize here, it's a lot of work, and I highlight some of the people that have done it, including Sonya Wolf, who's our next speaker. So the things that we see from these high interferon signals are increased dendritic cell activation. Especially after UV exposure of these keratinocytes, we see more cytokine production when keratinocytes are primed with interferons. We also have found that interferons really disrupt the skin barrier and promote colonization by Staph aureus, which can lead to more inflammation. And our group has also shown that these type 1 interferons inhibit Treg production after UV light, and so you actually don't get that negative inflammatory signal that you would normally expect after UV exposure. And one of the things that we think we're pretty confident in saying now is that we think interferons really drive this concept of photosensitivity. So this work was led by Mrinal Sarkar and Grace Heil, and what we found was that when we compared the cell death responses in lupus keratinocytes to UV light versus healthy control, the lupus keratinocytes always die more. So this is tunnel staining of our primary biopsies, and you can see that the death rate in lupus keratinocytes to low doses of UV, so about 50 millijoule exposure, which is about a half hour of sun in Michigan, so it's not that robust. You can see that about half the cells in culture will die with that type of exposure. And we can mimic this simply by overexpressing interferon kappa twofold in a healthy controlled keratinocyte population. And importantly, when we take our lupus keratinocytes and incubate them with an inhibitor of JAK1, which blocks signaling at the interferon receptor, we can turn off this really robust cell death response and make them look more like normal. So we think that this is actually an important potential treatment for blocking photosensitivity, and thanks to the NIAID Autoimmune Center of Excellence, we have a trial ongoing right now looking at JAK inhibition and how that affects photosensitive responses in patients. We think that the cell death that interferon is priming is actually an apoptotic response, and Shannon Loftus in the lab has done a lot of really beautiful work confirming that this is not pyroptosis and not necroptosis, but it's actually an apoptosis phenotype, and this is caspase dependent and probably caspase 8 dependent, and it seems to be related to IRF1, possibly upregulation of a protein called ZBP1, which I'll tell you about in a minute. So the other thing, so we think these interferons are really pathogenic. We think that they are promoting photosensitive responses, inflammatory responses, and cell death, but we don't understand why they're high. And so this is another question we've been trying to ask, is what's turning them on in the first place? So one of the things that we have seen, and this is lovely work done by Nazi Kermani in the lab, that STING activation is actually chronically on in this non-lesional, non-sun-exposed skin of lupus patients. So if you don't know what STING signaling does, here's a little brief tutorial here in the corner. So there's lots of ways for our bodies to detect nucleic acids, and inside the cell we have specific receptors that are able to recognize nucleic acids, and cGAS is one that will recognize double-stranded DNA. And when it finds double-stranded DNA, it will activate a protein called STING, which then binds to TBK, which then phosphorylates STING and also IRF3. And then IRF3 is a transcription factor to turn on interferon beta, interferon kappa, and other interferon expression in the cell. So when we look at our skin biopsies from patients, even without UV treatment, what we see is there's a lot of phospho-STING in the basal epithelium of these patients. So phospho-STING seems chronically on. And when you hit them with UV, you get a lot more. In healthy controls, when you hit them with UV, you can get some STING activation, but it's not nearly as robust as what you see in lupus. Also, just to highlight, Nazi has a poster, so go check it out. And then we think that some of this is coming from a particular interferon-regulated gene called ZBP1. And this work is driven by Ben Klein in the lab, and then Celine Berthier helped with this human data analysis. And just as a little primer, ZBP1 is a very interesting protein that's becoming very, very hot. There's like 5 papers a day, I think, coming out on this protein. But one of the functions of ZBP1 that's been recently described is that it's able to bind to cytosolic nucleic acids when they are in the Z conformation form. So I didn't know anything about this, but what it is that DNA can be in 2 forms, the B form, which is the normal double helix, and it can flip inside out when it's under either redox stress or has a lot of CG sequences. And when it flips inside out, it goes into the Z conformation, and it's not very stable there. But if it binds to a protein like ZBP1, it's able to stay in that Z conformation. And we know now, thanks to work done by Philip West, that the ZBP1 zDNA complex is able to then be recruited to cGAS and signal cGAS activation. So zDNA seems to be a very potent activator of the STING pathway. And I don't have time to show all the data from Ben, but what I wanted to highlight was that we have very clear evidence that ZBP1 is very upregulated in all of the photosensitive disorders that we've looked at. So we see high ZBP1 expression in cutaneous lupus, and it correlates very beautifully with the interferon scores in the skin. We also see it in childhood lupus, adult dermatomyositis, and also in childhood dermatomyositis as well. So we've looked at a lot of these photosensitive diseases, and we see ZBP1 upregulated in basically everyone that we've looked at. And we think what's happening is that in the context of UV exposure, that and again, I don't have time to show you all of Ben's data, but we think what happens is that when UV hits the cell, it causes a redox reaction in the mitochondria, which causes transient zDNA formation. And what we see is that if you increase ZBP1 expression by priming with type 1 interferon, what that does is it stabilizes the zDNA. And we can see that in In cells where you just treat with UV, you'll just see a tiny little bit of zDNA show up. When you prime with interferon, you see these really robust specks that form in the cytoplasm, and Ben's done a lot of work to show that these are outside of the mitochondria themselves, so they seem to be exiting the mitochondrial network. And we see that they co-localize with ZBP1 in the cells. And if we look at the zDNA, if it's also interacting with cGAS, we also see very lovely co-localization with cGAS. And when we look at the intensity graph, you can see that there's really good co-localization of cGAS and this zDNA after UV exposure, primarily after we prime with interferons. And if we look and compare healthy control and lupus keratinocytes and look at where the zDNA is going in the cell, again, after UV, you see some of this zDNA puncta forming even in healthy controls, but when you prime with interferon, first you see a lot, and when you have lupus, you don't need to even prime with interferon. They just really robustly upregulate the zDNA formation. And Ben's done work to show that if we knock out ZBP1, you can actually prevent interferon signaling, and zDNA seems to be a very potent stimulus of STING and activation of the interferon response. So I think I'm about out of time, but I just wanted to highlight a few, I think, key points. One is I think the healthy skin of lupus patients is really rich in type 1 interferons, and this causes it to be primed to activate other stromal and inflammatory populations, especially after UV exposure. And so when it looks fine, it's really primed for inflammation and isn't actually healthy. We think that this direct link between the skin immune activation and the systemic immune activation needs more research. And I just wanted to highlight some other people in my group that hadn't shown their picture yet that are working on some of this fibroblast myeloid cell crosstalk that we think that is really also highly educated and primed for by these interferons. And also Mitra Maz, who's an MD PhD student in my lab, has a poster, so go check that out. She's looking at the single cell RNA sequencing data from lupus and healthy control patients. And so in summary, what we think is happening in lupus skin is that in a chronic interferon environment, we're really driving a key regulator, which is ZBP1. And then when you get UV exposed in the context of high ZBP1 levels, this oxidized mitochondrial DNA, which is released to the cytoplasm, is stabilized by ZBP1 and causes hyperactivation of the STING pathway and upregulation of these interferon genes. And then work that Bin Zhu has done very beautifully has shown that these actually, the early interferon genes, including interferon beta, signal back and actually drive interferon kappa production, and we think that leads to a chronic loop in lupus skin. So we still have some work figuring out why the shutoff switch doesn't work, but I think we're pretty convinced now that this chronic interferon signaling is really important for photosensitivity. And with that, I will thank everyone that has worked with us. We have tons of collaborators, and I think a testament to the wonderful opportunities to do research here at the University of Michigan, and thank our funding sources, especially the Taube Institute, which has really funded all of our patient work. Thank you.
We'll have time at the poster session to ask some questions about Michelle, but we have time to take a question if someone has one. Yeah, go ahead.
Hi.
Skin regions that are not usually exposed to the sun, in those patients, do they also have a prime response to interferon, or maybe to address the chicken and the egg?
Yeah, it's a good question. So all of our data is from the question was, have we looked at non-sun exposed skin to see whether these mechanisms are still at play? All of these are from non-sun exposed. So, I mean, you can't guarantee that it's not fully not sun exposed, but we take it from the upper thigh. So unless they're like hanging out at nude beaches or something, most people aren't getting sun exposure there. So we've tried to like eliminate that. One of the things we're really interested in doing now is kind of comparing non-sun exposed to sun exposed normal looking skin in these patients to see whether there's any epigenetic switches that the UV is inducing, and maybe, priming this to, spiral out of control even faster. So I think there's still a lot of work to do, but yeah, really good question. Okay, thanks.
Thank you, Michelle. Our next speaker is Sonya Wolf-Fortune. She's a newly minted faculty member here at the University of Michigan in the Department of Pharmacology. She'll be talking on keratinocyte-macrophage inflammation axis in diabetic wounds. Sonya?
Hello, everyone. I'm very excited to be here today to talk with you about the role of inflammation in diabetic wounds. But before we can actually talk about the role of inflammation in a dysregulated context, we must understand its role during normal wound healing, right? Everyone in the room has experienced some form of skin injury. And following this injury, the skin will go through first homeostasis, where you have clotting at the site of injury and release of factors to help initiate the inflammation phase. During this phase, you have recruitment of neutrophils as well as macrophages with a pro-inflammatory phenotype that can secrete inflammatory cytokines, as well as engulf invading microbes and debris. You also have keratinocytes that can release cytokines and chemokines to assist in recruiting immune cells to the site of injury and manipulate their phenotype. And this phase is very important for being able to clean the wound to prepare it to go into a reparative state. And so towards the end of the inflammation phase, you end up having this switch from a pro-inflammatory macrophage to an anti-inflammatory macrophage that will then secrete anti-inflammatory cytokines and growth factors to help initiate the proliferation and reepithelialization phase to promote migration and proliferation of fibroblasts and keratinocytes. And this is going to be really important for helping to close that gap from the injury. And then following this proliferation and reepithelialization phase, you then have the resolution and remodeling phase, and this is going to be important for increasing the tensile strength in the skin. And so in the case of normal wound healing, inflammation ends up acting as a friend to help clean out that area to prepare it to go into a reparative state. But what happens when this phase is dysregulated? How then does this affect wound healing? One example of this is in the context of lower extremity diabetic wounds. This is a major complication for people with type 2 diabetes. And 70% of these wounds are unable to heal, and this unfortunately leads to amputation. And this is important because the 5-year survival following amputation for a diabetic foot ulcer is around 46%. And so this places a critical need to understand those factors that are contributing to the inability for these patients to heal in order to develop novel targeted therapies. One of those factors includes the presence of this delayed chronic inflammation. And this presence of delayed chronic inflammation ends up resulting in the inability of this wound to progress through the rest of the phases. And so in the context of diabetic wounds, this inflammation, because it's dysregulated, ends up acting as a foe and impeding proper wound repair. When we talk about the cell types that are contributing to this delayed chronic inflammation, a lot of research has been done to show that macrophage plasticity contributes to this by showing that in the context of diabetic wounds, early following injury, you end up seeing this decrease in pro-inflammatory macrophage phenotype and decrease in their secretion of inflammatory cytokines. And then late following injury, you actually see an increase in pro-inflammatory macrophages now instead of undergoing that switch like you see in normal wound healing. And they have increased production of inflammatory cytokines, and this contributes to that chronic wound environment. The macrophages are not the only cell type that can contribute to driving skin inflammation. Keratinocytes can also participate in this process. Maybe you feel like you're going through deja vu because Michelle just presented a lot on keratinocytes. And so they can do this through their immunoregulatory role, through their cytokine and chemokine production that can manipulate immune cells within the wound environment. But in terms of this role, this has not really been explored in non-healing diabetic wounds. And so I've been particularly interested in this aspect and understanding how these cells may be contributing to that dysregulation and inflammation that we see within the diabetic wound environment. In order to examine this, utilized human skin biopsies as well as the marine model for type 2 diabetes, which involves taking C57 black 6 mice, placing them on high-fat diet, And then after 12 to 16 weeks, they'll develop similar physiologic characteristics as that of patients with type 2 diabetes, including impaired wound healing. And then we can study wound healing by creating punch biopsy wounds on the backs of these mice and examining how they heal over time, as well as isolating out cell populations from these wounds to examine changes in their phenotype. And so overall, what we were able to see is that there's this dysregulation in the keratinocyte-macrophage axis within these diabetic wounds. And for the sake of time, I'm not able to go through all of the detail, but I just wanted to hit some of the highlights of what we've been able to see. So when it came to this early inflammation aspect, one of the things that we noticed was there was actually this decrease in interferon kappa within type 2 diabetic wounds compared to non-type 2 diabetic wounds. And this is interesting, as Michelle talked about, because type 1 interferon is interferon kappa is a type 1 interferon, largely produced by keratinocytes and is associated with driving skin inflammation. And we actually see it increased early following injury during normal wound healing, but it's decreased in diabetic wounds. And this becomes important because not only do we see that knockout of interferon kappa impairs normal wound repair, but it also, if you isolate out the macrophages early following injury, you end up seeing decreases in their inflammatory cytokine expression for those cytokines that are known to be important for proper wound healing. With seeing this importance for interferon kappa and regulating wound repair and seeing its decrease in diabetic wound, I became very interested with how it was being regulated within these diabetic wound keratinocytes, with a particular focus on epigenetic modification, most notably histone methylation. And so when I actually probed this area, what we ended up seeing was that mixed lineage leukemia 1 ends up regulating interferon kappa within these keratinocytes. And MLL1 is an activating histone methyltransferase. What does that actually mean? Well, it means that it can add a methyl group onto lysine 4, histone 3, and this can cause opening of the chromatin, making it accessible to transcription factors and turning on gene expression. And we actually see decreased MLL1 within these keratinocytes, and this is associated with that decreased interferon kappa that we see within these cells. Not only did we see dysregulation occurring early following injury, but we also saw this occurring late following injury and contributing to that increased late inflammation. In particular, we were interested in this increase in NLRP3 inflammasome activity that's elevated in type 2 diabetic wound macrophages that seems to be enhanced by the wound environment. However, the mechanism that's driving this enhancement has remained unclear. And interestingly enough, what we ended up seeing was that when we actually removed keratinocytes from these diabetic wounds late following injury and stimulated macrophages, we could see this drastic increase in NLRP3 expression as well as enhanced inflammasome activation in the presence of pathogen and damage-associated molecular patterns. And that this was being regulated in part through IL-1 receptor signaling. In order to understand those cytokines that were coming from the keratinocytes to drive this increase, IL-1 receptor can bind both IL-1 alpha as well as IL-1 beta. And when we examine these keratinocytes, while we didn't see very much IL-1 beta, we did see this increase in IL-1 alpha. For example, this is examining our human single-cell RNA sequencing data focused on the keratinocyte population, and we see this increase in IL-1 alpha expression within these type 2 diabetic wound keratinocytes, and we also see it in the murine model as well. And this is interesting because IL-1 alpha is constitutively expressed and induced by keratinocytes following injury and is also associated with driving skin inflammation. And interestingly, if we actually stimulate these macrophages with IL-1 alpha, we can see this increase in NLRP3 expression within these cells. And when we probe for the regulation that is occurring here, we ended up seeing that JMJD3 seems to be regulating NLRP3 expression within these macrophages. And so JMJD3 is actually an activating histone demethylase, which means that it can remove a methyl group from lysine 27 on histone 3, and this can cause opening of the chromatin, making it accessible to transcription factors and turning on gene expression. And so we end up actually seeing increases JMJD3 within these diabetic wound macrophages and downstream of the IL-1 receptor signaling, and it seems to be regulating that NLRP3 activity. So through probing this keratinocyte-macrophage axis, it's been interesting in revealing possible therapeutic targets, for instance, showing that restoring of cytokine presence, such as interferon kappa, if you do it early following injury, that this not only increases macrophage pro-inflammatory phenotype, but it also seems to improve diabetic wound healing, as well as blocking receptor signaling. So if we block IL-1 receptor signaling, this seems to result in decreased NLRP3 activity in diabetic wounds, as well as if we block epigenetic enzyme activity in a cell-specific manner. For instance, blocking JMJD3 in macrophages not only seems to decrease their NLRP3 activity, but it also seems to improve wound healing. And so overall, when we talk about inflammation during wound healing act as a friend or foe, it really does depend on the context. So you see normal wound healing, it seems to act as a friend to clean out the wound, but in the context of diabetic wounds, because it's dysregulated, it acts as a foe and impedes proper healing. And then when we examine the factors contributing to this chronic inflammation, we see this dysregulation in the keratinocyte-macrophage axis. And this has revealed possible therapeutic targets for further investigation to develop treatments for these diabetic wounds. And so I think when we think about future directions in this area, I think continuing to probe this immunoregulatory role of keratinocytes in these wounds will help lead to novel therapeutic targets. But not only that, but understanding the inflammation pathways within this setting will not only help with wounds, but also with other skin inflammatory diseases associated with diabetes. That includes psoriasis. And so doing further investigation in that area. So with that, I would like to thank all of the members in the Gallagher Lab. JD Moon has a poster presentation, go check it out, as well as my collaborators and all of my funding. And with that, I'll take any questions. I don't know if there's time.
So we do have time for one question. Yeah, Vince?
Yeah. Great talk. Great to hear it again. Question is, NLRP3, all of these responses are often in response to bacteria, which we think of as quote unquote pathogens usually. And for all the times I've treated diabetic foot ulcers and given broad spectrum antibiotics, I always get worried that what we're doing is we're actually shifting the signaling. Do you have any evidence that perhaps some of the bacterial signaling is actually beneficial in trying to resolve this, to try to have the right friendly immune responses opposed to the pathogenic ones?
I think when it comes to the bacteria response within the wound and the diabetic setting, it seems to be polymicrobial, right? And so it seems to be more inflammatory, but you're right, by sometimes maybe activating those responses slightly like NLRP3 during normal wound healing, it's not as bad because it's able to help in regulating that inflammation early to help clear out the wound. But then if you're activating it chronically in the diabetic wound, it may not be as beneficial because now you're having that chronic release of inflammatory cytokines. Thank you.
Thank you, Sonya. So our next presenter is Professor Beth Moore. She's been a longtime immunologist here, a huge international reputation, and will be speaking on immunopathogenesis and immunity in the lung. Beth?
Yeah, thank you so much. It's my pleasure to be here. I was charged with trying to tell a story a little bit about friend and foe with about 5 or so 6 minutes with each of those stories, so I will do my best. So if you missed the punchline, what I'm going to try to do today is tell you a brief story, admittedly both of these incomplete, about how neutrophils can directly help promote fibrotic lung disease, but then how the milieu of the fibrotic lung can then impair the host defense function of neutrophils. So the disease that my lab has been interested in for over 2 decades now is a disease called idiopathic pulmonary fibrosis. It's the most common interstitial lung disease. It's sadly progressive and ultimately fatal for these patients. And what it's characterized by is interstitial thickening, the deposition of extracellular matrix like collagen, and it stiffens the lung. And so these patients have trouble breathing in and they have trouble exhaling as well. The origin, as the name implies, is unknown. But we know that this disease is associated with chronic infiltrates, and I'll show you those can be both macrophages as well as neutrophils. We know these patients are highly susceptible to infection, and that sadly, infection really worsens the prognosis of their disease. So there have been many studies, I just show you some work from us, but there have been at least 15 studies in the literature that have associated increases in neutrophils in circulation with worsening outcomes in patients who have pulmonary fibrosis. So this is work from our group that looked at 133 IPF patients, just characterizing their baseline levels of neutrophils in their blood at their clinic visit as being above or below 5,000 per microliter, and then looking at their disease course over the next year. And I hope you can appreciate that those who had the higher levels of neutrophils in the blood in blue have a worse disease outcome, and those patients were succumbing and had poorer survival. To be able to do anything mechanistic, it's very hard to do that in humans, and so we've moved to a mouse model. Admittedly, not a perfect model of IPF, but it is a model that develops fibrotic lung disease. So this is bleomycin as a chemotherapeutic agent. If you instill it in the lung on day 0, it'll give an acute lung injury phenotype and an inflammatory response for, say, the first 7 to 10 days. And then that will gradually turn into a fibroproliferation response, and you'll get fibrosis, usually peaks around day 21. You can see on the bottom down there that we can use an assay called a hydroxyproline assay. That's a modified amino acid that's uniquely found at about 33% in collagen, and so you can measure that as a rough surrogate for collagen deposition in the lung. We usually get about a doubling of hydroxyproline in this model. On the right, you can see there at the top, those are saline-treated lungs with nice normal architecture, easy for the mouse to breathe and exchange gas. But you can see 21 days after bleomycin that you've got lots of consolidation, lots of inflammatory infiltrates, and on the bottom, that's a trichrome stain where the blue coloring is collagen deposition. And if you look inside at a little bit higher magnification, you can see that there are a number of neutrophils. There are macrophages as well, but I'm only telling you the neutrophil story today. All right, so in this mouse model of bleomycin, what you can see is if we just take the whole lung and do a bronchoalveolar lavage, we just squirt saline in the mouse lung and we pull it right back out, and then measure the number of neutrophils that we can see. I hope you can appreciate that while there's almost no neutrophils in a normal mouse lung, that at day 7, 14, or 21 after bleomycin, you can see that they increase and then they stay elevated. And just as a snapshot, you can see that in the blood on day 21, you can still see elevations of neutrophils in the circulation of these mice that develop fibrosis. And I'll call your attention to the scale. In this model, in the BAL, we get about 10 to the 4th or so neutrophils. If we were to look at the whole lung digest, we'd be in the 10 to the 6th number, and that'll come back of importance in a minute. So one of the things we noticed in these mice that were developing fibrosis is that if you look in the BAL where we had these neutrophils, you can see evidence of sort of activated phenotypes. So in that first panel, you're looking at neutrophil elastase, which is an enzyme that can be secreted by activated neutrophils. The next panel is myeloperoxidase, another activation marker from neutrophils. The next panel is looking at extracellular DNA. That could either be netosis or it could just be alveolar injury and dying epithelial cells. But in the last panel, we're looking at citrullonated histone H3, and that should be a marker relatively specific for netosis. So it seems like there is activation of these neutrophils in this lung. And so we wanted to ask the question of whether the fibrotic lung environment was actually causing these neutrophils to undergo netosis. And so to do that, we just collected that BAL fluid from mice that had either been treated with saline or with bleomycin on day 21. And we took that, that lung milieu, that cell-free supernatant, and put it on top of neutrophils from a normal mouse in a dish and asked whether that induced netosis. And so what you can see here in that first bar, that's just the media control with very little netosis measured here as a cytox assay for extracellular DNA. Ionomycin's the positive control, so you see a nice induction of netosis. And then the saline fluid, the fluid you could get out of a saline-treated mouse had really no activity for netosis above baseline. But you can see that the fluid that you can get out of a fibrotic mouse could really induce netosis. And while there are many stories that I could tell you about how netosis can promote lung injury and damage, I'll just show you this one piece of data. This is actually not work from us. This is work from the Greek group. And what they showed is that if they collected the nets that could form, either from neutrophils that they just left in culture untreated, or from the neutrophils that they could harvest from bleomycin-treated mice, and then put that net material on top of normal lung fibroblasts, in the untreated there, you see there's no induction of smooth muscle actin. With the neutrophils from the controlled neutrophil nets, no induction of smooth muscle actin. But when you put the fibrotic nets on these fibroblasts, you see nice induction of myofibroblasts. And the reason myofibroblasts are important is that we know from lots of work, some of which was done here by Simfan, that these myofibroblasts are actually the producers of the extracellular matrix that is really pathologic in this disease. And then in that bottom panel, I hope what you can appreciate, in the green is alpha smooth muscle marking the myofibroblasts, and in red is the elastase that would mark the neutrophils. And so you can see that a lot of times these co-localize in the lung. So we really think that these neutrophils, they come in, they degranulate, they damage, they go into a net formation, they're right on top of the fibroblasts, and then they can really promote the development of this extracellular matrix. All right, so that's at least the story about how neutrophils can be bad. But if you remember, I told you the lung of these fibrotic mice are full of activated neutrophils, and yet these patients are very susceptible to infection, and they in fact get recurrent bacterial infection, and that seemed like a conundrum to us. And so I'll tell you that about 10% of IPF patients that are hospitalized at any one time are being hospitalized for a bacterial pneumonia, and they have very high 30-day mortality rates if they are hospitalized for pneumonia. This is a study that looked at the kind of pathogens that those patients get, and while you may not be able to read everything, I can tell you that Streptococcus pneumoniae and staph species make up more than half of all of the bacterial infections that they get. And because this is a talk about neutrophils, I'm going to tell you about MRSA, because MRSA is the second most common infection that these patients get, and it's supremely dependent on neutrophils to be cleared. So it's a good assay of neutrophil function in these patients, or in the mice. All right, so to do this, what we did is we went back to our mouse model. We gave the mice bleomycin or saline on day 0. We let them develop a fibrotic response in the lung. And so then on day 14 or on day 20, we then gave them a MRSA infection. And we just asked, do the saline-treated mice handle MRSA better than the fibrotic mice? In the bottom on the left here, what you can see in the open bars are, we put about 10 to the 7th MRSA into the lung. That's a lot, but it takes a lot of MRSA in a mouse model. You can see that from 10 to the 7th installation, that the normal mice, the saline-treated mice, are clearing it. There's about a 3-log reduction in their bacterial burden 24 hours later. For the fibrotic mice, either at day 14 or day 20, there's about a 2-log reduction. So they're definitely clearing, but they're not clearing as well. And importantly, if you ask whether the mice can maintain that infection, can they hold it in the lung and be able to clear it in the lung, they cannot, because we can see evidence of sepsis in these mice because the bacteria get out of the lung and then go to the spleen, and you can see the increase in the CFU in the spleen that we can see at day 21. So these fibrotic mice are not able to handle this MRSA infection. They do worse, just like the patients. So you can imagine our surprise when we said, okay, well, let's just look at the inflammatory response. And the first thing we noticed is that it seemed like there weren't enough neutrophils in response to the MRSA infection. So this is where the numbers become important. If you remember, I told you that there were about, maybe 10 to the 4th to 10 to the 6th extra neutrophils in the lung in the fibrotic mice. But when you give a mouse a MRSA infection, they need to be able to recruit 10 to the 8th level neutrophils to be able to handle that infection in the lung. So what you can see there is that in the saline-treated mice, they bring in, somewhere in the 60 million or so neutrophils into the lung, whereas the bleomycin-treated mice couldn't mount that additional response that they needed. Even though they had a high baseline, they couldn't generate the incredible extra neutrophil response that they needed. So the reason that they can't generate that response is because the fibrotic lungs are impaired in their ability to make the chemokines that actually recruit neutrophils. And so neutrophils express CXCR2 as the chemokine receptor. They respond to CXCL1 and 2 as the chemokines that recruit them. And I hope you can appreciate on the right-hand side there that if we look for the ability of the lungs of those mice treated, saline or bleomycin treated mice with MRSA to be able to make those chemokines, it's reduced in the fibrotic mice. We still don't know the mechanism of this. That's something we're working on. So then the question became, well, maybe it's just a numbers problem. They just can't get enough neutrophils in there. So what if we could force them to get enough neutrophils in the lung? Could we help them? This idea being we might, recruit neutrophils to the lungs on purpose to help these patients when they see an infection. So now we gave the fibrotic mice CXCL1 to just recruit more neutrophils before we gave them their MRSA infection. So what I hope I can show you here is that in the first line there, you see the number of neutrophils recruited for the saline-treated mice in gray. In blue, you see the reduced number of neutrophils from the fibrotic mice. But if we give them CXCL1 NEC, we can now recruit the same numbers of neutrophils. So now we've got fibrotic mice that have a lot of neutrophils in their lung, the same number as the normal mice, and yet they still can't clear their infection. So what that means is it's not just a game of numbers, it's also a game of function, and that means that the neutrophils in these fibrotic mice are actually impaired in their function. So I will tell you, to make this story short, that it's not phagocytosis. The neutrophils from the fibrotic mice are able to engulf the bacteria, but they are not able to kill the bacteria that they can eat. And so this is a killing assay that actually accounts for the number of bacteria that are actually taken up by the neutrophil. And if you look at surviving bacteria in blue there, you can see that the fibrotic mice can't kill the MRSA that they're able to ingest into those neutrophils. And I'll just remind you that if we think about what are the mechanisms that are responsible for innate immune cells to be able to clear MRSA bacteria, it's really a story about reactive oxygen species. So as the phagosome is taken up with the MRSA inside of it and the phagolysosomes fuse, you should be able to generate reactive oxygen species that will kill the MRSA and then be able to clear that infection. So we next wanted to look at the production of reactive oxygen species. And again, in those first two lanes, you're looking gray bars are saline, blue is bleomycin. At baseline, there's a little bit of a lower level of sort of basal reactive oxygen species, but you're not making a lot of reactive ROS until you actually need to stimulate that. You can see in the next gray bar that when you give those mice MRSA or give those cells MRSA, you can see this big induction in ROS production in the normal cells from the saline-treated mice, but from the fibrotic mice, that induction of ROS is impaired. And similar to the same experiment that we did to look at netosis, we just asked whether the milieu of the fibrotic lung could impair the ROS generation and impair the killing of these neutrophils. And the answer to that is actually yes. So again, looking at bacterial survival here, you can see in yellow is just the media control. In green, we use TGF-beta, so for the fibrosis aficionados in the room, you'll know that TGF-beta is like one of the most well-described pro-fibrotic cytokines. It would have been such an easy answer if it had been TGF-beta, but it's not. TGF-beta does not impair the function of neutrophils. Saline, fluid from a saline-treated mouse does not impair their function, but fluid from the lungs of a fibrotic mouse that's acellular can definitely impair the function of these neutrophils. And again, this is still an active area that we're trying to understand exactly what this factor is. So does it matter? So I want to tell you that I've already told you that patients who get a bacterial infection do much worse with this disease. But interestingly, if you just go and you look at the basal microbiome of patients who have lung fibrosis and you sequence it, you can see that some patients have higher levels of staph and strep species in their lung. And if you stratify those patients by sort of their basal levels of staph and strep species, you can see in the blue bars there that the patients that have the higher loads of bacterial DNA associated with those pathogens, really we don't even think of pathogen because these patients don't have pneumonia. It's not risen to that level. But patients who carry higher loads of staph and strep in their lungs actually have much worse outcomes as well. So I hope what I have convinced you is that the fibrotic lung, not only does it seem to have structural defects that might contribute to its infection, but it also seems to have innate host defense defects so that there are two ways that these patients actually become more susceptible. And I hope that our future goals are really to try to understand the soluble mediators that influence NETosis and the immunopathology side of this process, and then the impaired bacterial killing as well. So with that, I want to just say this is the work that was done by Helen Moore-Niemi, who's a graduate student there in the middle with the big arrow, but also helped by David O'Dwyer for the clinical samples for the microbiome work, Xiaofeng Zhou, and then Gabby Intrup, you'll see in the pants there. And Gabby has a poster on a completely different project related to Pseudomonas infection and obesity, poster 6. So please check out Gabby, and thank you so much for your attention.
We have time for a question?
No.
And again, at the wine and cheese end of the day, we'll have time to chat with the speakers more.
Got it.
Beth, hi, great talk.
Hi, Euless.
I know it's in the lungs and the oxygen tension is really, really high, but does oxygen tension alter the ability to generate reactive oxygen species?
Oh, good question. I don't know the answer to that. I've only ever, I don't actually know the answer to that. I don't even know if there's literature on that. Good question. I don't know. Hi. Pierre.
Beth, I really enjoyed this. But I'm interested in the issue of comorbidity. So if at the height of your bleomycin phenotype in the lung, have you tested in other tissues whether their ability to recruit neutrophils, if challenged, was normal? Or are you, by that time, are you dealing with a systemic disease?
We have not done a lot of challenges outside of the lung to look at that, but what I can tell you is that even the neutrophils that come from the bone marrow in those mice are impaired. So we do think that this local lung injury does lead to signals that at least tell the bone marrow to alter the neutrophil production. And since you're a neutrophil aficionado, what we get are aged neutrophils instead of the more immature, the younger neutrophils. And so those aged neutrophils are a little bit more prone to sort of these NETosis phenotypes and things like that. So we definitely see that shift in the chemokine receptors and all. So it's as if the bone marrow is like shoving out these old neutrophils, and then those neutrophils can't get back into the bone marrow to be cleared. So the whole homeostasis of the neutrophils is off in those mice. Thanks. All right, thank you.